Welcome back, everyone. In this lesson, we'll explore the fundamentals of experimental design, randomization, control, and replication. We'll also see how to carry out random assignment in Excel and analyze results in Jamovi. By the end, you'll understand how strong design leads to valid, reliable conclusions. Experimental design is essential for valid science. It prevents bias, reduces confounding, and helps us make accurate, reproducible conclusions. Poor design can waste time and lead to false claims, so getting this right matters. There are three major pillars. Randomization prevents selection bias by giving every subject an equal chance of being in any group. Control keeps conditions consistent and isolates the effect of the treatment. Replication repeats treatments on multiple subjects to estimate natural variability. We also often add blinding to reduce unconscious bias. An experimental unit is the smallest entity that gets a treatment. This could be a person, plant, animal, or sample tube. A treatment is the condition we apply, like a medication, a fertilizer, or a temperature level. You should define these before collecting data to avoid confusion later. There are two common design structures. Completely randomized design, everyone is randomly assigned to groups. Randomized block design. First group similar subjects into blocks, then randomize within each block. Blocking helps control for known sources of variation. Let's practice random assignment. Enter your subjects' names in column A of Excel. In column B, type equals rand to generate random numbers. Sort the list by those numbers. Divide the list into treatment and control groups. Save as CSV so Jamovi can open it. Next, we'll analyze in Jamovi. Open Jamovi file, select your CSV. Go to the Data tab. Set group as nominal, a factor, and outcome as continuous, a scale variable. This step helps Jamovi apply the correct statistical tests. To explore the data visually, go to Exploration Descriptives. Move outcome to variables and group to split by. Under plots, select box plot and means with 95% CI. This quickly shows how groups differ and the variation within each group. To test for differences, if you have two groups, go to t-tests independent samples t-test. Put outcome as the dependent variable and group as the grouping variable. Check normality and variance assumptions. Add effect size, Cohen's d. If you have more than two groups, use ANOVA one-way ANOVA. Put outcome as the dependent variable and group as the fixed factor. Check effect size and run Tukey post hoc tests if significant. When finished, you can right-click any table or plot, choose copy or export. Save outputs as images or PDFs for lab reports or presentations. Always connect your results back to the design. Randomization, control, and replication make these results trustworthy. When you run real experiments, document exactly how you randomized subjects, what your control group was, and how many replicates you used. This level of detail increases the transparency and credibility of your work. A few common problems to avoid. Not randomizing introduces bias. Too few replicates reduces power. Poor or missing controls leads to ambiguous results. Confounding variables hide the true treatment effect. Now it's your turn. In groups, review a short study summary. Identify the experimental unit, treatment, control, and design type. Then discuss. Did the design follow the principles we covered? We'll share out as a class. Remember, good design makes stronger conclusions. Use randomization, control, replication, and blinding. Tools like Excel and Jamovi make implementing and analyzing experiments much easier.